They cause about $1.5 billion of yield losses every single year in the United States, and it's a growing problem. Mothers kind of like get into the roots, they form these cysts, and they die with their eggs inside of them. And with the type of exudates that are being excreted, it kind of allows other pathogens like sudden death syndrome to um, affect the plants. So
Mm.
we're seeing a ton of yield loss every single year from these guys. And it's hard to treat. They're really hard to identify. It's hard to tell if you even have them in your soil.
Oh.
And then they're hard to treat because soybeans, for example, are a row crop, so applying pesticides aren't really efficient. And there's also other reasons why we shouldn't be just spraying pesticides.
Niah is an aspiring mycologist with a strong background in fungal research. Hands-on experience, we cover so much of her past research and also her future research, current research in cultivation and foraging. She works with Bigfoot Mushrooms, so she also grows her own mushrooms, and we talk about farmers market talk, helping to grow and sell a variety of gourmet cultivated mushrooms while maintaining her own at-home cultivation. Niah's passion extends beyond the mushroom itself, though. We talk about the soil food web, how she's deeply interested in agroecology, and how fungi can support healthier crops and more sustainable farming practices. So yeah, we cover a lot of good, juicy tidbits in this episode. She also explores a powerful role fungi play in protecting delicate ecosystems like wetlands. So yeah, we cover her current stuff with wetlands and then her past stuff with Plant Path. And also she just created an Instagram, so please go follow her. Dirty Hands Plants and Fungi. I will post that down below. All right, let's get into this week's episode. Let's go. Thank you for being my first guest in the new house. I'm very excited about this chat.
My name's Niah. I'm an undergraduate researcher. I also work on a mushroom farm, sell at markets, big into foraging, and all sorts of stuff, fungi. I'm fortunate enough to have grandparents who are into foraging.
Oh, cool.
I'm so grateful for that because I think for things like foraging, that's generational knowledge that's passed down. And I was so close to just missing out on that. And I think a lot of people are missing out on that generational knowledge being passed down. And so I'm just very grateful that my grandma Reen and grandpa Jimbo got me into that. And yeah, then I started working in plant pathology and realized that like a lot of these techniques I can do at home and can transfer these skills, and just really got into the world.
Mm-hmm. Yeah, I want to hear more about your research because I feel like not a lot of. I really am interested in plant pathology. I want to go back and get my doctorate in that. So I'd love to kind of pick apart your brain a little bit on like, what does that include? Like, what is plant pathology and what kind of research are you interested in? Mm-hmm.
So, plant pathology is the study of plant diseases and plant pathogens. I'm personally really interested in agriculture. I really loved working in a plant pathology lab. I'm working in a biogeochemical lab right now, and I
Mm-hmm.
love it too. But I think I'd love to get back into plant path more.
Mm.
Um, so we looked at a lot of stuff with soybeans. I live in Ohio, so soybeans are pretty big here. Um,
Yes.
I was specifically looking at soybean cyst nematodes and looking for a fungi that was antagonistic against them to
Okay.
treat for them instead of using like uh, insecticide or pesticide.
Mm-hmm. Yeah, what are nematodes? Can you maybe define what those are if people aren't familiar with those little things?
Yeah, nematodes are really cool. Uh, Nematodes are a type of arthropod, so like a tardigrade. They're like little round worms, microscopic, and there's probably around a million different species. I want to say there's a lot. There's a lot,
Whoa.
and they're hard to identify. So they're kind of split into two categories. There's the free living nematodes. Those are the ones that eat like microbes, plants, or bacteria, fungi, that type of stuff. They really help keeping the bacteria in check in our
Got it.
soils. Um, they also help a lot with decomposition. They don't directly decompose materials, but they break down some of that organic living material.
Right.
And then there is the other set of nematodes, which are the parasitic nematodes. So
Ah.
those are defined mostly by what they eat. They're really hard to identify. So we kind of just classify them by what they host on.
Mm.
So there's stuff that, um, you know, parasitize humans. Like we have like hookworm, pinworm, things like that, like other mammals.
Yeah.
Um, there is nematodes that attack insects, like there's a specific nematode that attacks the fig wasps and
Mm.
parasitize those. And then there's a whole set of nematodes that parasitize the roots of plants, which are what soybean cyst nematodes are, and what I was interested in at the time.
Yeah.
Um,
Okay.
so soybean cyst nematodes, um, they're a hugely economically important pest. They cause about $1.5 billion of yield losses every single year in the United States, and it's a growing problem. Mothers kind of like get into the roots, they form these cysts, and they die with their eggs inside of them. And with the type of exudates that are being excreted, it kind of allows other pathogens like sudden death syndrome to um, affect the plants. So
Mm.
we're seeing a ton of yield loss every single year from these guys. And it's hard to treat. They're really hard to identify. It's hard to tell if you even have them in your soil.
Oh.
And then they're hard to treat because soybeans, for example, are a row crop, so applying pesticides aren't really efficient. And there's also other reasons why we shouldn't be just spraying pesticides.
Mm-hmm.
But yeah, so I was interested in finding a fungi that was antagonistic against the nematodes.
Okay. Okay. And what did you find?
So I started off by taking some soil samples in a Ohio soybean field that did have a high count of nematodes.
Okay.
And then I went through this crazy process of high throughput isolation of fungi. Took like 9 months of isolating fungi. I was having dreams that I was a plate of fungi. It
Not having dreams about it. Oh, that's
was a
awesome.
lot of work. It was a lot of work, but at the end, I had about 650 isolates,
Okay.
which that's a pretty big collection. And now on the backside of that, doing other research, I'm realizing like it's so cool to have those fungal collections just easily accessible in your lab. Um, yeah. So after that, I kind of did some Sanger sequencing, some PCR work, which going into it when I first got hired at the plant pathology lab, I didn't know anything about PCR. And if you're a home mycologist and you're looking to get into PCR and that type of stuff, I would highly recommend. I don't think it's as hard as like the perception of it is. I think
Mm.
that anymore, like the materials and stuff that you can get are pretty affordable, pretty easy to understand, and yeah, you can find labs that'll sequence your stuff for you for pretty cheap now.
Right. Absolutely.
But
Yeah.
yeah, it wasn't my favorite part of the process. My favorite part of the process was actually the bioassays that I was doing. So I was trying to filter down through those 650 isolates to get the ones that are actually antagonistic. So I did
Right.
um, I wanted to look at I needed to find a fungi that both produce chitinase and protease. So mushrooms are
Mm-hmm.
made of chitin, and so are nematodes. So we would expect most fungi to produce chitinase in some form. Um, and these assays were so cool. So I took like a petri dish with agar that had chitin in it, which kind of made it more opaque.
Okay.
And then I put my isolate in, and when the fungi grew, it started breaking down the chitin in the plate. So I would get these rings of like clear agar around the fungal growth, and that's how you could tell if it was producing chitinase.
Right. Oh, yeah, that would make sense.
They were
Huh.
really pretty. They were really fun to look at. Um, and and then finally, my last step to kind of filter these fungi were what I called the nematode fighting rings. So I
Okay.
grew some soybeans, inoculated them with some soybean cyst nematodes, and then once they formed the cyst, I kind of created a ring of cysts around the fungal growth in the middle, and then saw like how they interacted with the fungi.
Ooh. Yeah, because I was gonna ask, you were saying earlier that it's hard to identify like nematodes in the soil. So how are you kind of figuring that out? Is there like something you can just analyze of like how many microbes are in a soil sample, or is there like a specific nematode test?
So we had like purchased the nematodes to inoculate the plants
Oh,
with, and then you can see the cysts on the roots. So
oh, I see, I
the
see.
nematodes form these cysts. They're
Mm-hmm.
called like soybean cyst
I'll have to
nematodes
look that
because
up.
there's other nematodes that form cysts like that and parasitize plants. And that's what is like how you're going to identify them if you have them in your field.
Got it.
Unless
Okay.
you're out there, paying someone to do soil testing and stuff like that.
Mm-hmm.
Um, but yeah, so got the cyst to form, collected the cyst, and then did the little fighting ring. And then unfortunately, this was the part of the experiment where I had to like leave for some personal reasons. I chose to take a break from school for a little bit.
Mm-hmm.
Um, so I had to leave this research behind, but this fungal collection is still being used, and I think there's definitely potential for some of these isolates to be processed and turned into some sort of like bio pesticide,
Mm-hmm.
something like that.
Yeah. Yeah, absolutely. And so you were doing that research. So what are you kind of working on currently then?
So I took a little break from school
Mm-hmm.
and realized how much I loved research. I loved being in a research lab. Um, so I started up at Kent State University and work right now in a biogeochemical lab. Like I said, we do
Mm-hmm.
a lot of wetland ecology work. Um, so fun, so much fun, and like just like a different, like completely different path to go down
Yeah,
compared to
agriculture
pharmacology.
and like wetlands are very different, but maybe there's some similarities with stuff that you work on in both.
Yeah, for sure. Um, so I was from the start, from the first day of me being hired, I was like, how do I create a research project with fungi in these
Ooh.
wetland systems? Um, so in Ohio, H2 Ohio's main focus is to protect Lake Erie's watersheds. So Ohio's an ag state. We have a ton of agricultural runoff that contaminates Lake Erie and causes things like harmful algae blooms,
Mm-hmm.
which can affect our drinking water.
Yeah.
So wetlands really help filter some of this nutrient runoff. So I know in other states, wetlands ecology focuses on other aspects of why wetlands are so beneficial. But here in Ohio, I think a lot of our research into wetlands is mostly for nutrient budgeting.
That's a good point.
So
All about
yeah,
money.
yeah. So plants actually take up a lot of nutrients, and I think they're overlooked a lot of times in nutrient budgeting because once they break down, they're just releasing all those nutrients back into the
Mm-hmm.
environment. But fungi really help plants uptake more nutrients, and then once they do decay and break down, they help recycle that nutrients and get it back into the system
Mm-hmm.
a lot faster.
Yeah.
So that's kind of a project I'm designing and working on right now. I'm kind of looking at how fungi can help plants uptake nutrients.
Okay. Especially in
Um,
like wetland kind of areas. 'Cause I would imagine if it's really wet, how are they really connecting as much? Or is that a small-minded idea?
no, that's a really good point. So
Let's take a little breather. Yeah, this is a really nerdy episode. Hopefully, y'all are loving it. Hopefully, we broke it down a little bit, kind of like fungi do in the soil. But I just want to shout out to today's sponsor, which is me. Flora Funga Podcast is sponsoring today's episode. And I wanted to chat with you about this new tincture that I am providing you. So we got the Flora Funga with Flying Fungi Tincture. This one is called Calm and Collected. I honestly find that this sleep time or nighttime tincture, or if you're just trying to have more of a relaxing time, the Calm and Collected really, really helps. So we got reishi as the mushroom, and then the plants in here, because obviously it's Flora Funga. You gotta have both. You can't have one without the other. We got reishi, ashwagandha, and lemon balm. So this three species tincture, very, very tasty. Honestly, it's one of the best tinctures I've ever tasted. and also really helps me just wind down at night. So I just take 1 to 2 milliliters. If you've never taken a tincture before or an extract, you just kind of plunge the top, throw it back, just let it sit there for a few seconds, and then swallow. It's really good. It tastes really good. It's me just kind of sleep through the night. DM me the word calm, and I can get you hooked up on this new tincture. So thank you also Flying Fungi for making this product. I will be actually venturing to his mushroom festival that he will be having in New York coming up this fall. So hit him up if you want to get some more details on the mushroom festival that he's hosting in New York. He was talking about how there's no mushroom events up in New York, so he's gonna start one, and he has invited me to kind of do my thing there. So if y'all are interested in meeting up and going to this awesome mushroom festival in New York, please DM me about it or DM him about it. So thank you, and sorry I didn't want to take up too much of this episode, but I wanted to give you a little breather in between. So all right, back to this week's episode.
Fungi's role in the environment really depends on a lot of different environmental factors. So you might have a fungi that is acting beneficially in one way, but then because wetlands are they fluctuate so much, they could become inundated, and then if it's super wet, then those fungal connections aren't as strong.
Right.
So that is a really good point. These systems, wetlands are so complex, and we don't know that much about the fungi that are in them. And that's the more and more research I do and reading I do, the more I'm like, wow, we don't know anything.
Yeah. Yeah, absolutely. I took a wetlands course, and that was just like one of my most eye-opening things because there's so many different definitions of different wetlands. And I had a really good professor, and so he was very strict on like what classifies as like, one wetland from another. And so it's just like, yeah, learning the different factors of like what that costs. And then yeah, the different ins and outs, like inlets, outlets, very complex. So that does sound really interesting. But we never learned anything about fungi within wetlands. So I'm like, there's probably so many other things to uncover. So I'm glad you're doing that.
Yeah.
Mm-hmm.
And it's been fun. I'm really grateful to be able to do the type of research I'm doing. Like as an undergrad, just
Mm-hmm.
kind of the support system I have at Kent in regards to research is really great. I have a lot of good mentors and people around me to help me out with my stuff.
Cool. I don't know if we can go back to agriculture, but I guess I kind of wanted to. I was reading some of your ideas of what questions, and I want you to explain what trap culturing is. I don't know where that really plays a role, if that's more of the other one or the wetland. But yeah, what
No,
is
for sure.
trap culturing?
So trap culturing is a really cool method that researchers use to multiply arbuscular mycorrhizal fungi.
Okay.
It's used to help identify spores, establish monocultures, and make custom inoculums. So when I worked in Plant Path, I did 9 months of high throughput isolation. That was a lot.
Okay.
I really didn't want to do that for this project that I'm working on now. And also with arbuscular mycorrhizal fungi, which are the fungi that associate with plant roots, they really need to be on like a living plant. So they're hard to just work with in a lab setting. Trying
Yeah.
to buy cultures like that it started to get just too much for the scope of my project. So I kind of developed this trap culturing method that has been used in other research projects to create my own little custom bioinoculum to use.
Oh, so you kind of came up with this process.
Not, I wouldn't say that. I wouldn't
Okay.
say that,
You're
but
just kind of
um,
like the main user of this technique, maybe?
yeah, I think that like a lot of researchers use it for like obtaining spores and doing monospecific cultures.
Got it.
My how I'm using it is pretty like general use. I think that like the actual method itself can be used in agriculture too,
Okay.
because what it is, it's I collected wetland soil, and then I'm growing a host plant, which I chose clover. And so the roots of the plants are in the soil, all of the microbes are in the soil,
Yep.
and then the AMF, which I'm looking at, are going to travel up into the roots of the plants. And they can harvest them, chop them up, and then use the roots and a little bit of the rhizome
Hmm.
to create kind of a a biofertilizer.
Oh, okay. Yeah, that was another question I wanted to ask is like, how are you kind of incorporating this, your past research, or the agricultural topic into this type of fungi-based product? Okay.
Yeah.
So how does that exactly work if if you're taking like the rhizomes from that? I want to learn more.
Yeah. So the rhizosphere and the roots are going to contain the AMF.
Mm-hmm.
Once I chop them up, I'm going to kind of inoculate another wetland plant, soft rush, with the custom
Oh.
inoculum. And then I'm going to have like another set of sterile plants, which, the sterile soil is supposed to be my control. I think it's important to note that plants don't grow in sterile soil. So it's a little bit of like you know, it's not the best control system. I've been playing around with the idea of like a microbial wash to kind of balance it out and have like a small controlled community of microbes in both experiments.
Mm-hmm.
And then the second layer to this research is I'm going to do a high phosphorus treatment and a low phosphorus treatment, which we
Oh,
can get into why that is later. But
okay.
um, yeah, I think trap culturing is pretty cool. And I think there's a lot of applications for farmers to use similar methods and just in general, caring about your soil microbes if you're a farmer.
Yes.
Um, the best management practices that you hear about are the best management practices for a reason. And yeah, they have a like they benefit fungi the same way that they will benefit your plants. So
Right.
reducing your tillage, reducing the compaction of your soil, um, reducing the amount of fertilizer you're using. So,
Right.
really honing in on your fertilizer efficiency. Because if you're over applying fertilizer, you're not going to have those fungal associations.
Mm-hmm.
Not applying a bunch of pesticides and crap to your soil, not using a bunch of monocultures, using host plants, rotating your crops, things like that. Things that we know are good for agriculture are also good for fungi.
Yeah, absolutely. And that's cool that you were using clover as like was it like your testing type of plant
It's a
or
host plant. So that's like the
I see.
actual trap culturing plant.
Okay, got it.
So yeah, the
Okay,
microbes are going to hang out in the roots of the plant and then I'm going to chop them up and then they're going to go in a second set of plants.
cool. I like the visuals. I'm a visual learner, so I liked all the
Thank you. Yeah, I feel like I've tried to explain trap culturing to so many people and they're like, "What?" And
You're
I
like,
had
wait,
to
wait,
like
okay,
draw some
this
diagram. Yeah, yeah.
it's perfect. I was like, okay, there's the roots. I got it. Have you
Yep.
heard of mycorrhizal applications?
Yeah.
What's kind of the difference with like that trap culturing or a different type of product versus that application?
Yeah. So I think the mycorrhizal applications, they're pretty cool. I think that they definitely need some more development and research.
Mm,
And same thing with my work in plant pathology. I think that these fungal solutions are going to be crucial in the next 100 years of agriculture in this country.
hmm.
You know, by 2050, we're going to have another 40 billion people, 40, yeah, 40 million people to feed on the low end. That's a lot more people to feed.
That's a
So
low end. Oof,
yeah, yeah. So we
yikes.
need to start. I think agriculture is key. I think that's where the focus needs to be.
Hmm.
Um, and I think fungi can definitely help. But one thing to consider is that like the microbes in my field are going to be a lot different than the microbes in your field, even if we're neighbors.
Yes.
So
Yeah, definitely.
it's hard to create a fungal product that is going to be applicable in everyone's field. And then, like you were saying with the wetlands, there's so many environmental factors that really affect what role microbes play in the environment.
Right.
So it's microbes are so complex and I think trying to develop a product right now with the knowledge we have is hard. I've done some sub experiments using like the happy frog soil, like the potting soil that has all of the different
Yeah.
microbes in it and yummy stuff, and
Hmm.
it seems to really help the plants, especially compared to like sterilized soil. Plants don't like growing in sterilized soil, it turns out.
No,
um,
no, that makes sense. There's nothing to help it. Or I guess it's all hurting it, but yeah, there's nothing to help it at
yeah.
least. Yeah, and
Yeah.
having a good variety of different, because yeah, you were right with like the different soil, and then you have different concentrations of like bacteria and fungi, and because like here in Florida, our soil is all sand, but yeah, in the Midwest, it's like you got so much more better organic material. It's like a darker soil, so yeah, we'd obviously would need different things. So that's actually where mushrooms come into play here too. We have a lot of our spent blocks and make our own compost with that, and then you can put that into the garden beds. Way more processed, way more money to like make like living soils, but then, it attracts whatever plants that you're growing. So then fruit trees are better here versus it's all about the plants. What does the plants really want also?
Yeah.
Oof, so complicated.
Yeah. The spent mushroom substrate stuff is so cool. I work at a mushroom farm and we just like produce so much of it, you know?
Yeah.
We just have so much of it around. And it's, we throw it in our little field and we sell it to some local people, but
Mm-hmm.
there's a lot of mushroom farmers who I think just like don't know what to do with all of their mushroom substrate. So if you have a little home garden and you're like, man, I want a cheap and easy solution to like help with water retention, help with like not having to put as much like fertilizer into the soil, like hit up your local mushroom grower. They'll probably give it to you for free, honestly. Like
Yeah.
they don't know what to do with it either.
Yeah, it's usually about like moving it is the hardest part. Like just come by and pick some up. But yeah, the house that I just moved into, I just realized that they have their own like composter back there. Like it's like a whole thing where you can like change the ventilation, and I was just like, "Whoa, I didn't even know this existed." So I'm excited to actually like have a real compost because I've never been able to have that. Just like something in your apartment and you're like, like what do you do with that if you can't really throw it anywhere? So yeah,
Yeah,
I'm really excited for that.
that is so exciting. I've tried to
Mhm.
do the apartment compost bins. It doesn't they don't work out very well.
Especially if you don't know where to bring it to, you're just like, all right. Don't know
Yep.
where to go. This episode is sponsored by Gaia's Garden Goods. We're here to just give you a little fun fact of the day. So this one covers lobster mushroom, Hypomyces lactiforum. This is a really fun fact. Not a lot of people know. A lot of people do know about the lobster mushroom, but did you know the lobster mushroom isn't a single mushroom per se? It is hijacked by another fungi, a parasite fungi. When hypomyces infects a russula, or a lactarius mushroom, it grows a dense crust around the outside skin of the mushroom. Two things happen here. It remodels the host shape, so russula versus a lobster mushroom have different shapes. So the parasite wraps around the mushroom and changes the gills, changes the shape. It also rewires the flavoring, so like the full chemistry of it. Hypomyces brings its own metabolism, changing the compounds of how it smells, taste, and texture, turning something that's pretty bland or inedible now into an edible mushroom that people forage all the time. That's where you get the seafood like smell or taste with a lobster mushroom. It's just because of that parasite. Without it, it would be a Rousselot or a Lactarius. If you want more fun facts, make sure to follow and subscribe. And thank you guys, Garden Goods.
There are fungi trapping nematodes and there are nematode trapping fungi.
Ah, okay.
So they kind of keep each other in check, I think.
Okay.
So it's cool. They have like the fungi have these little like lassos. They like lasso
Yes.
the nematodes, bring them in, and I think they like excrete something to like break them down.
Ah,
I'm sure it's
so I
protease and
see.
nicotinase and that type of stuff.
I'll have to find a video or something because I remember learning this in like a plant class somewhere. But it was just like, yeah, like the lasso effect, and then just like, oh, that's such a fun visual. Like, I want to see this fungi lassoing
Oh
some like
yeah,
nematodes, trapping it in, breaking it down.
yeah.
Wow. Okay.
No, I think the synergy between like all of the soil microbes is so interesting. It's food webs above ground are really complex, but I think the food webs in the soil are at least 10 times more complex. Like
Yeah.
it's so interesting to me. We just know so little.
Yeah, I
We
think
know
that's
so
what it
little
is
about it,
too.
and there's so much to learn. Um, I think organic farming is pretty cool, but like
Hmm.
a lot of organic farmers don't know what's going on in their soil. They're kind of just like messing around and seeing what works. So I'm really interested in finding out like what specific species, what specific groups are doing that work. With my trap culturing stuff right now, it's pretty general. It's just like, is this little bioinoculum I made gonna work or not? But if I got the money to do some like more sequencing work, that would be really cool to figure out like, oh, what did I even trap in this? Like, what's going
Yeah.
on in the soil?
Hmm.
Um, yeah, there's a lot to learn.
Yeah, yeah, absolutely. Yeah, the below ground communities are just so unique, and yeah, we don't even probably have the names to some things. I don't even know. Like there's bacteria to actually moving little creatures to yeah, different kinds of fungi. Wow. Yeah, there's so much to learn. And that's why I like this kind of field too. There's just so much to do and learn, and I'm glad people are really interested in helping it. And the soil is kind of where I think we're gonna help the planet the most because that's how we're growing all of our food. And if we don't have nutrients in our food, then we don't have nutrients. So I don't know why it's taking so long to figure out, we got to heal the soil first. Hmm.
Yeah, yeah. It's hard
Absolutely.
because the systems we have in place, we've relied on for so long in agriculture.
Hmm.
Um, it's hard. Like I said, we're gonna have to feed a lot more people in the next 25 years. And I just don't think the direction we're going is sustainable. The nematode problem is like getting worse and worse
Hmm.
because of our practices, because we're
Uh,
using contaminated farming equipment, because we're planting the same thing every single year, because we have these monocultures. There's
Dang.
a lot of things to do, to work on and develop. I think that agriculture is the key, and I think that having relying more on your local communities and your local farmers is really cool. I've been privileged enough to sell at the farmers market in my local community, and talking to the farmers and just experiencing like showing up every week and being able to talk to people in my community about like how great mushrooms are, how they're so beneficial for their diet and everything
Yup.
is just like it's so fun. It's so fun. I love it so much. And it's hard work. Like I really appreciate the market farmers, and
Hmm.
if you're frequent at your local farmers market, I want you to know that like those farmers appreciate you.
Yes.
Plants can have two ways of taking phosphorus. They um, can do it themselves. They use their roots and their root exudates and other acids to uptake their phosphorus. Um, plants are pretty good at this. They've been doing it for a very long time.
Hmm.
Um, but the issue is that there's limitations to what they can do and how far the roots can reach. So a lot of times you'll have like P depletion zones around the plant roots,
Hmm.
where there's just low phosphorus levels. And that's where the second pathway comes in. There's also the mycorrhizal pathway, and the mycorrhizal fungi also have their own root exudates. They have their own transporter proteins, and they're able to reach a lot further into the soil. They have like a lot finer, fibrous, like roots.
Yeah.
Um, they help the plants uptake even more phosphorus, which again is kind of what I'm looking at in my research project.
Hmm.
Um, the really cool thing about this, though, is that it seems that there's some sort of fungal signaling going on. So if the plants are using one pathway, the other pathway will kind of like turn off. Mycorrhizal fungi are providing nutrients to plants. The plants are also giving the fungi something in return. So
Right.
if there's a ton of phosphorus in the soil, the plants are like, oh, I'm chilling. I can collect all my phosphorus. There's no reason for them to associate with the microbes. Yeah, the soils become depleted. They will reach out to the fungi and have them help them out. So
Hmm.
there is a little bit of like predicting you can do with what type of pathway plants are using. Um, my lab, like I said, does a lot of nutrient budgeting, and some of the methods for nutrient budgeting I don't think take into account these like specific pathways. Um, it's hard to calculate what how much more phosphorus the soil can hold. So,
Hmm.
just looking at what specifically affects those things, I think is really important. And So yeah, that's kind of the reason why I'm having these two phosphorus treatments in my experiment. Um, my hypothesis is that in the high phosphorus treatment, the plants aren't really going to associate with the microbes as much. But then in the low phosphorus treatments, I think that they will associate a lot more. And then
Mm-hmm.
after I grow them, I'm going to do some like plant tissue digestions and figure out like specifically how much was taken up and then compare the differences between the four treatment types.
Okay. And are you looking at other plant nutrients or mostly just kind of phosphorus? Because that's
Yeah,
main one.
I'm mostly looking at phosphorus. I think for me, I'm an undergraduate and I just the scope of my project every day is getting more and more outside of undergraduate research level work. And
Yeah,
so I really need to like hone in and just like pick a couple of things to look at.
yeah.
I just want to like learn and know everything and test everything. And there's so many aspects of this experiment where I've had to be like, okay, that's super cool, can't do it. Like identifying
Yes.
what specific species I'm culturing and trapping into the roots of my plants, I would love to know. But like ultimately, I need to graduate sometime and there's only so much funding. So
Yeah. Yeah, you're definitely sounding higher up than undergrad at this point. I feel like yeah, you're juggling many things, but also you're just like inquisitive and that's awesome too. Hmm.
I appreciate that.
Absolutely. And have you found that maybe climate change has been affecting the nutrients or even maybe the increase of nematodes in general too? Or is it mostly our practices, I guess?
I would say it's mostly our practices. I think that like um, you know, there's a very strong connection between wetlands and agriculture.
Mm-hmm.
And, you know, we are applying crazy amounts of fertilizer every single year to our fields, and not a lot of that is actually being used by the plants.
Yeah.
So a lot of that gets pushed into the wetlands and into the waterways, and even contaminates our drinking water streams. So
Mm-hmm.
um, yeah, I think it's mostly our human practices that
Okay.
are affecting them.
Mm-hmm. And also, do you think our human practices are creating this golden oyster that's kind of getting maybe out of control in the Midwest? I know a lot of mushroom farmers are actually halting growing the yellow oyster. Do you think that, I mean, that plays a role, or what's your opinion on the golden oysters?
So I do have a controversial opinion on this.
Ooh.
Um, I will say that, I feel like I'm somewhat knowledgeable in the realm of mushrooms and,
Mm-hmm.
know a little bit about ecology. This is my take, and if you have a different take, that's, completely fine.
Absolutely.
I've been seeing a lot of headlines this past year about how golden oysters are an invasive species and it's the cultivator's fault. It's the cultivator's fault. And I see this at the market. People like come up to me and they're like, "It's your fault." Like, yeah, and
Whoa. It's you.
okay, okay. So the problem is, if you're not familiar, um, golden oysters are grown commercially, and the spores escape the lab, and now we're finding them all over. And I've seen this myself, especially this year in particular. You go out into the
Yeah.
woods and find these huge clusters of golden oysters, and to me, I'm like, great, that's my dinner. Like happy as
Boom,
can
more to
be.
eat.
But the issue here is that like they're kind of outcompeting other oyster species that are like native to the area.
Mm-hmm.
So, on a biodiversity aspect, I think this is a problem. However, I think that these type of headlines are kind of harmful because one, there is other invasive species that are actually causing environmental harm beyond just biodiversity. We're creating rifts between cultivators who are providing nutritious, delicious food for the communities and customers. Again, biodiversity is something we should focus on and like strive for, but golden oysters are providing benefits to the environment. So I
Hmm.
think calling them an invasive species is really kind of harsh. I've kind of settled on the term non-native,
Yeah.
although I feel like they would have ended up here anyway. Spores travel. Like they're, you know, I'm
Yeah.
sure I'm sure cultivators made the problem worse, but
Hmm.
I don't know. I think that these types of headlines are kind of harmful, and you know, if you're really worried about the issue, my advice is you go out into the woods and you hunt them down and then you eat them. And
Boom.
then, like, it wouldn't be a problem.
Problems up. You could solve all the issues. Yeah, I feel like sometimes it's like sprinkling salt in the ocean. Like, it's already there.
There's a lot of different groups of mushrooms that are doing some pretty cool things in our
Mhm.
environment. Armillaria is pretty interesting to me. I
Okay.
wouldn't say that's like a misunderstood mushroom by any means, but I think that like some of the harm that they cause trees, I think they're helping keep tree populations in check and help breaking down some of those materials in the woods. Yeah, I think decomposition processes are really cool.
Mm-hmm.
Microbes do a lot to help like push those processes along. And then in regards to phosphorus cycling, decomposition acts as both a source and a sink,
Mm-hmm.
depending on a lot of different environmental factors. So I think we just need more community studies. We need more just more people into mycology. There's so many things out there to learn. Yeah, I'm just a nerd for it.
Yeah, I love it. You're a perfect guest on Flora Fungal Podcast because we like to get down and nerdy. So it's perfect. Where can people find you or follow you on social media if you have any of that?
Yeah, I just recently started an Instagram. It's
Yeah,
called 30 Hands Plants and Fungi.
love
Um,
that. I said love
Hopefully
that.
be at MSA this year if you want to find me
Yes.
there.
I think I'll
Um,
be there too.
nice.
Well, I hope you enjoyed this week's episode with Niah. Love talking about her research. I just love getting down and nerdy with her. She's really fun to talk to. Very soothing voice if you noticed. But yeah, if you are interested in being a sponsor on Flora Fungal Podcast, please hit me up. If you want to support the podcast, I have a GoFundMe site, so you can just find all of that juice right here. If you also want to join my Patreon, so shout out to all my Patreons for helping me, the podcast, and also you get early release stuff, behind the scenes adventures, additional content. So please sign up, patreon.com/florafungapodcast if you want to sign up as little as, $1 a month, all the way up to really supporting me with like $25 or $20. So that's kind of the highest tier. And you get a free t-shirt. So if you do sign up for that one, you do get a free t-shirt. So, all right. Well, thank you for enjoying this week's episode. Comment down below what you learned or maybe something that you found really fascinating. But yeah, and if you want to try the tincture, the Calm and Collected, please DM me the word calm or just hit me up via email. All my handles are Flora Funga Podcast. Go follow my new YouTube channel. We're trying to get the backup one still, so we're still working on that. We're serving YouTube. But yeah, the backup one is called Flora Funga TV. So feel free to follow that one as well. I'm up on Rumble, Patreon, Instagram, LinkedIn. You name it, baby. I'm on there. All of my stuff is also up on my website, so that's a good way to contact me as well. florafungapodcast.com. All of the episodes are up there, all the videos are attached, and all of the audio is embedded, so you don't even have to download a third-party app to listen or watch. Your beautiful host, KK. All right, have a great week, my scientists, and go learn something new today. Peaches!
I hate golden oysters.
Crush them!
Love figs. When I got this shirt, I was like, oh my gosh, it's so perfect.